Good afternoon, everybody, and it's my pleasure to welcome you to the first of our NeuRAtalks webinar series for 2022. I'm Carole Renouf. If we haven't met before, I'm the Executive Director of NeuRA Foundation. We'll be sending you a follow-up email tomorrow to alert you to our next NeuRAtalks in May, which will be on resilience, well-being, and mental health. And I invite you to join us again and share the registration link with family, friends, and colleagues. If you've attended these before, then you know that I aim to structure the session so that I have around 25 minutes to talk with our panelists, followed by around 20 minutes for you to ask your questions. Please don't wait until I throw the floor open to post your questions in the Q&A feature. The chat box is disabled, and please don't raise your virtual hand or you'll distract me. Just use the Q&A along the way, although we won't pick up your questions until later in the piece. A quick reminder that we don't have any doctors on the panel today, so we're not equipped to answer any questions about someone's specific condition, prognosis, or treatment. But as far as research goes, I'm privileged to have a terrific lineup for you. 2 doctoral or PhD candidates and 1 postdoctoral fellow, and they are all incredibly intelligent, articulate, and driven. Our theme for today is future leaders. So we're going to delve into their experience of a research career and doing their PhD, as well as into their particular areas of expertise. One of our passions and responsibilities at NeuRA is nurturing the next generation of scientific and clinical leaders. And take my word on it, you don't get better than these 3. So let me introduce them one by one. Hayley Leake found fame in 2021 by winning Australian Survivor. But as tough as that was, she will tell you that pursuing a successful research career is even tougher. Hayley is a physiotherapist by training and is about to submit her PhD on chronic pain in adolescents. Dr. Steve Kassem studied neuroscience and psychology originally, but has become an explorer and navigator of the brain, along with his mentor, Scientia Professor George Paxonos. Steve also has the unique distinction of having modernized the Golgi stain. We shall hear more. And finally, Kerith-Rae Dias is a couple of years into her PhD in neurogenomics after doing a Bachelor of Science in genetics and a Master's of Forensic Science. Her passion is translating genomics into healthcare, and her PhD focus is intellectual disability. Let's speak with each of them in turn. So Hayley, I'll go to you first, and let's start with the Resolve clinical trial at NeuRA. You were the clinical lead on this, and I know you can't share the results yet, but give us an insight.
Yeah, hi Carole, thanks for having me here today. So the Resolve trial was really exciting. It was a big trial run through NeuRA, headed by Professor James Macaulay, and it was really looking to find solutions for chronic low back pain, which is a huge problem. Low back pain is the leading cause of disability globally, so definitely ripe for finding new solutions. So the intervention had 276 participants, 2 arms to it, and the whole point is trying to deliver this novel treatment that really focused on the role of the central nervous system. It had a 3-pronged approach. So the first part was really looking at pain education. So there's a lot of research saying if we can reconceptualize pain rather than being this construct that it is always damage to the body, but reconceptualizing it as the brain leading pain and sometimes being overprotective, how can that help? And then it had 2 other elements that were really vital that had to do with retraining the sensory components to the skins with some tactile training for the lower back, and then a graded integration back into movement to really precision functional training for the body. And we've had really, really promising results. And as you say, yeah, we are under review and it is not yet accepted, but it is very close with the journal. But I can say, yeah, it was really clinically meaningful results and we can't wait to share them all.
It sounds great. As a back pain sufferer myself, I'm looking forward to it. Okay, let's get the survivor question out of the way, shall we? How did you deploy your knowledge of the body and how the brain determines response to pain to win?
Yeah, it was, Survivor is quite a segue from the PhD. It was a very interesting conversation with my supervisor when I was like, I'm going to step away from the PhD for a minute to do a reality show. But it actually came in quite handy, having a really deep understanding of the body and especially the science of how pain works. Survivor has some really long challenges in them that can be painful. And there was a few key ones that I ended up winning. And I think my pain knowledge really helped there. Albeit having some pain in a long challenge is not the same as chronic pain, but I could still apply the same principles. So particularly this idea I would ask myself when I was standing for 5.5 hours on these tiny pegs that was really, really painful for the feet. I would ask myself, okay, this is painful, but is it dangerous? And that's a key question that we get people in pain to ask themselves, because we're trying to reframe this concept that pain always means damage, which isn't true. It can be protection, it can be overprotection. So I asked myself that a fair bit in the challenge and allowed myself to really reframe the idea that this is going to cause any problems. It was painful, but I felt very confident that 5.5 hours later, I could step off those pegs and I would be okay. And that was true. And I won the challenge. I ended up winning the show.
Congratulations. It was a terrific achievement. On a more serious note, let's go to your PhD. I'm really interested in the topic chronic pain in adolescents. I guess firstly, because chronic pain isn't something we would normally associate with adolescents, right?
Yeah. A lot of the time people will say to me, I didn't realize that young people could have chronic pain. It is surprising because we do think of it as this problem that happens when we're older, but it's not true. The estimates we have right now put the prevalence of chronic pain in young people between 11 and 38%. So it's quite high. And it can be such a disabling problem for a young person, because adolescence especially is such a critical stage of development. Young people are engaging in different kind of social constructs and learning about the world. They're at school, and when they have chronic pain, a lot of those things are taken away, and we see kind of these ripple effects later into their life. So finding better ways to treat chronic pain in youth is really important.
So a PhD requires you to make a unique contribution to the field, right? So how are you approaching that?
Yeah, so my PhD has looked at some of those concepts I talked about in Resolve, which really centers around helping young people make sense of what chronic pain means. How does the science of pain work? What is happening in your body when you're experiencing chronic pain? There's good evidence in adults that reframing that can lead to better outcomes, but there is nothing really in the pediatric space. So I've been working with a variety of people globally to try to adapt some of those principles to young people. We can't really just take the science that we have in adults and apply it and give it straightly to kids. They're a totally different kettle of fish. And so it's really applying, taking those principles and trying to figure out, how do we teach young kids about how pain works? When we do that, does that help?
Okay, got it. And for you, I know we spoke a bit about this. What have been some of the hardest aspects of doing a PhD?
Yeah, lots of the PhD is tricky, but that's intellectually stimulating and interesting. I would say the hardest thing has been the self-motivation. It is a really tough thing because you are very independent in your PhD. The best thing you can do, which I feel very lucky that I had this, is surround yourself by excellent mentors, have an amazing space to work in, and other great colleagues to work with, other PhD students. That has been key, but COVID took a lot of that away from us, and I've had 2 years of the PhD with a lot of work from home. So really continuing to self-motivate when life keeps throwing curveballs can be really tricky. But for me, the key has been having a really good workspace with people who can adapt to a digital technology and continue to support each other.
Thank you very much, Hayley. I'll turn to Dr. Steve Kassem now. So Steve, I know you are an explorer and innovator by nature. Why don't you start by telling us about the Golgi stain? What does it do and why was it important to modernize it?
Well, it's probably considered one of the most important stains in neuroscience. So Carole is sort of considered the father of neuroscience. It's over 100 years ago, he developed this stain, and it was the first one where it could stain the entire neuron, the brain cell, in its entirety. Not just the cell body, but all its arms and all its tentacles to go and talk to the other neurons. It was the first time we could do this, and it was extraordinarily important for that and promoted decades of research. But by about the 50s and 60s, it started to get pushed to the side because it was so incompatible with a lot of the more modern techniques. It wouldn't work with other things we were doing with the brain. And so what I did, and part of my explorer nature, I suppose, is I loved looking back and reading these old works. And so I was reading his original works from 1890, 1901, and I realized there was room to modernize it, and there was room to go there to do, some minute changes and some other changes to temperature and things. And I could actually apply that stain now with modern techniques and then improve it as well. So now it's almost like a flat photo on glass of this black outline of the neuron in its entirety, something we couldn't accomplish before. And I suppose something I'm quite proud I'm able to use again, and something I plan to use in my future work.
I think you should feel very proud of it. Let's focus on your PhD now, which was a little while back between 2013 and 2017. And I know when we spoke, you shared with me some pretty dire personal experiences you had during that time, something to do with mercury poisoning, as I recall.
Well, that's right. I suppose it reminds us to be passionate as well, right? You've got to have a passion for it. And my passion might have gotten a little over the top when I was doing some of my work. I'd worked 72 hours straight at one point because I was too excited to stop. And I'd lacerated myself, I'd lesioned myself and spilt mercury onto me. And it actually got absorbed into my system. And so I didn't realize until after the next day when I had crippling pain in my leg and I couldn't quite move it properly. I've gone to the doctor and he's gone, "Have you been exposed to any heavy metals?" And I'm like, "Well, I did spill some mercury and some other things on me the other day." And he's like, "Well, I think you've got mercury poisoning and it's starting to atrophy your leg." And so I've had to go through, I had to take 6 months off. I had to use a cane. It didn't stop me though. I could still read. So I was doing research at home and I was still able to keep up my job on the weekend. But there was more. I mean, I look back on it with fun now. It's a fun little story to tell, but I had a stroke within my family that I had to look after as well. I had my first weekend at the age of 27. So I was either working or studying or contributing to my PhD in some way, or ill, or dealing with family issues. But it shows that it gives you the strength you need sometimes. It shows you where you need to focus. And you can always discuss with your supervisor, "Look, I need some time here and some time there." And if your passion and enthusiasm continues to go through strife like this, which will happen, it validates that you should complete your PhD.
I think we should have you on our next NeuRAtalks on resilience. But anyway, so today you work very closely with George Paxon, as we said, on mapping the brain and providing clinicians and scientists around the world with atlases. How unique and meaningful is this work?
Well, just to start with, it's extraordinarily meaningful to me because I didn't know George was Australian. I was an undergraduate and I'm using his books as a student. And I'm seeing the word Paxonos, and I'm like, what an amazing man. And I've just happenstance to realize that George is actually here in Australia at NeuRA, the world authority on neuroanatomy. There's no one more expertise than him. And so the meaningful very much to me, I brought the book I stole from the library as a student with me the first time I met him. But on a more broad sense in the research, there is not a single paper that wouldn't either cite George's work or have reference to George's work in another way. And so the atlases we make, these map the anatomy, the connections, and all the other details we need to know about the brain in humans, but he's also done it in animals. So the humans are self-evident. We need them in surgery. We need them in human studies. But by doing it in animals as well, we can make these comparisons, these analogies between things we can do in the animal that we can then extrapolate to the human. And that's where a lot of work has really exploded in neuroscience, because we can obviously explore things we can in an animal that we can't in a human. And having these linked up, it's extraordinarily significant. And he's earned his title as one of the world's most important neuroscientists. And I'm privileged to be with him to contribute to this.
If I'm right, I think together you guys have discovered a number of new regions of the brain. But let me just ask you one final question then. I know on one occasion you spent 40 hours in the magnetic resonance imaging machine having your brain scanned, which has to be beyond the call of duty. But why was that important?
Well, it was important because we couldn't find any subjects. No one wanted us to go in there for 40 hours, and we needed to do this research. So I volunteered. I didn't do 40 hours at once. They wouldn't let me. So I had to split it over 10 different sessions of 4 hours, which was the medical maximum you could do. And it was so important because we needed that many hours in the machine to get the best image we could possibly get of the human brain. And people have, and our lab has as well, looked at post-mortem brains where we can scan them for as long as we want. But the problem is after someone's passed away, the brain changes. It changes its shape. It starts to deteriorate within moments. And so by able to do this in a living human, we can not only get the best representation of the brain how it is, but we're approaching the image quality that we couldn't get before by having me sit in there for 40 hours.
I think I remember you telling me you managed to go to sleep. So
I was lucky.
All right, I'll move us on to Kerith now. Kerith, fascinating background. You were born in India, you grew up in New Zealand, you worked in London, you ran the Kinghorn Centre for Clinical Genomics at the Garvan. Then you decided to undertake your PhD while on maternity leave with your second child. What drives you?
Thanks, Carole. I have to say, all that moving around definitely makes it difficult to pick your favorite cricket team. So, but I think there are two parts to your question. And on a very personal level, I think like most of us scientists and researchers, we're inherently curious about the world around us, and we're also motivated to learn from failure. I have to credit my parents and my early upbringing in India for instilling a really strong work ethic. On the PhD front, I'm highly motivated by the real families that we work with. So one of the projects I'm working on is with a cohort of individuals with intellectual disability who've had no answers for years as to what is causing the disorder. And through this study, I'm also finding new genes related to ID and modeling the disorder in human stem cells and mini brains to understand the mechanistic basis of these genes and their link to ID. The new technologies in genetics are revolutionizing diagnostics. And myself, my supervisor, Tony Roscioli, and the rest of the clinical team at Prince of Wales, we're very driven not only to use these technologies to make a case, to bring them to the clinic. We want to help finances for these families, but also, I guess we are able to advance the understanding of the biology of ID.
That sounds amazing. In terms of your experience with the families, I know you told me that the genomics of intellectual disability are highly variable, and many parents go on I think you called it a diagnostic odyssey for 8 years on average before they get a diagnosis, and 50% of them actually never get one. What are the consequences of that for them and their child?
Well, that's right. And for the families, and particularly the child, it means moving from clinician to clinician. It means multiple misdiagnoses, unnecessary and sometimes invasive tests, taking time off work and school, incorrect management, and sometimes incorrect treatments, but also huge costs. It's been estimated that the social and healthcare costs for a family with ID is around $200,000. So we know that the more severe forms of ID have a genetic basis. So sequencing the genome of an individual has a huge benefit as a first-tier test. So the New South Wales government has recognized this, and now eligible individuals can have trio exomes. So that's where a child and both of their parents, their exomes are sequenced, and this is rebatable under Medicare. So this means that 50% diagnostic rate is increasing with every family that is tested.
That's really interesting to me because I have a sister with intellectual disability, so I know firsthand what it's like, you know, growing up in a family in that situation. You also told me, I think, that a lot of the genetic variants that you're looking at in intellectual disability are not inherited. And that would mean a lot, I think, to parents to hear. Is that part of what you're trying to achieve?
Absolutely. Part of my research is cataloging all of the genes that cause ID, and we're now well at over 2,000 genes involved, and this makes ID very heterogeneous, so it presents differently in different people. The main form of mutation or variation in these genes that cause ID is de novo, and what this means is that that variant is not inherited from the parents. So when a de novo variant is found, this undoubtedly gives the parents a huge sense of relief that they haven't contributed to their child's disorder. But more importantly, because we've given them that information that this variant cannot be inherited, they can plan for more children.
Yeah, I think I mean, I know in my own family, the issue of parental guilt was a big one. So yes, terrific. Okay, let me focus more on now your startup as part of your PhD. You had the opportunity to set up a startup. Tell us about that.
Yes, I think I really wanted to explore the full, journey that comes with doing a PhD, not just the research aspect of it. I was very excited about that prospect. And I worked very closely with the clinicians at the Prince of Wales Hospital, and I could see that there was an opportunity to speed up variant analysis, both with data mining and machine learning techniques. So I signed up to UNSW Founders. They have a program called NeuRA for women founders specifically. I let them know of my idea, and I was able to attract a team of 3 other incredible women with software development and business skills. So we're very proud of the team because we came second out of a group of about 80 women. We're currently iterating our prototype, and we're applying for seed funding. So fingers crossed.
All right. And to finish again on the PhD, my last question for you. So our audience is probably not aware that most of the PhD students at NeuRA come with a government scholarship, which is terrific, but it is worth about $28,000 a year. You obviously have a partner, but you're doing your PhD full-time and have no other income. We provide a top-up to our PhD students. Can you give me an example of how useful that is to you?
Extremely. I love living in Sydney, but we're definitely paying for it. So when I decided to undertake a PhD, one of the first chats with my partner was about finances. At the time, we had 2 kids in daycare full-time and a mortgage, and I didn't want the PhD to add to any financial stress. So when I met with Peter Schofield, he mentioned the NeuRAtopup scholarship, and I think it definitely helped in my decision to choose to do my PhD at NeuRA. It helps towards living costs in Sydney. And I think one small but important for me example that I can showcase how that's helped me make decisions is I buy a PhD parking permit at UNSW, and it's not cheap, but what it allows me to do is it allows me to be flexible with my work hours, because a lot of the time I'm juggling around my kids. I work nights because of that as well. And it allows me to safely come in for when I need to do cell work or culture work at night. So I know that a lot of my peers don't have that privilege, and I'm very grateful for it.
Yeah, that makes a lot of sense. I understand how important that would be. Okay, I'm going to throw the floor open now to questions from our audience. There's a good one actually that's come in for all 3 of you, so I might call upon you each in turn. As I've got Kerith there, we'll go with her first. What's one thing that would have made your life easier going through your PhD?
Oh, that's a really good question. A really good support network is essential. We don't have family here that can help with the children and things like that. So I think for anyone thinking about doing a PhD, I would highly recommend having a really good family or friend support network that can help with that. I know that that's something that's been integral to me doing my PhD.
Okay, I'll go to Hayley next.
Yeah, it's hard not to mirror that because that's so important, having your own support network. I think one thing that's been really vital for me is the conversations I had with my supervisor to try to find the right match before I started. It's really important that the topic is valuable to you, but the relationship you have with your supervisor is so integral to your journey and the success you have during that. So I would credit that, and that's my big advice to anyone who's looking to start a PhD, is to get that relationship right.
Good tip, Steve.
For me, it's sort of on the nose, is that I didn't have a scholarship. So a scholarship for me would have made my life a lot easier. But of course, I have to echo everyone else. I mean, I obviously had a support network that permitted me to go through what I already talked about. But I didn't have the money to do it. So I had to work weekends and evenings and all the rest of it. So mine's sort of the inverse. A scholarship would have been extraordinarily helpful.
Okay, thank you. Steve, I might stick with you actually, because there's a question specifically for you. What excites you about the development of technology like very high-res MRIs for the advancement of brain mapping?
For me, it would be just like an astronomer gets a new telescope, or if NASA gets the new Kepler, we get to explore and see things we weren't able to see before. So in a similar way, this technology gives us new telescopes and new technology to see what's in our minds, things we haven't seen before. And we still discover new parts of the brain. You talk about, oh, you can't possibly be finding new parts in the brain, but we do. I've already found 3. I've got another 57 to go to catch up to George.
That's wonderful. I refer to it as the Uncharted Brain. Yep.
Absolutely.
All right, thank you. Here's one for Hayley. Hayley, how did you get into pain research in the first place, given that it's quite a specific field?
Yeah, great question. I came through and did my physiotherapy degree in Adelaide, and I was offered the chance to do an honours project, and I had a list of options to choose from. And one of those was a project on chronic pain that looked really interesting to me. But what looked also very interesting is the fact that I would have the opportunity to work under someone called Professor Lorimer Moseley, who was at NeuRA, and just like an amazing mentor, an excellent communicator. And there was so much I wanted to learn about research from him. And the more I got into that path, the more I just saw the need in chronic pain for there to be better treatments. I worked clinically for a while as a physiotherapist, and I could see me and my colleagues were struggling to apply things that would have good long-term outcomes. So it was a combination of the support that I wanted to have for research and the clear need for there to be some better treatments.
And actually, to your point earlier about your supervisor, I have a question asking specifically how inspirational James Macaulay has been for you.
James, yes. I don't know how I got so lucky to be surrounded by such incredible people in the space. James has been an excellent in just bringing together a really clever and diverse team of people that are there to lift each other up. I think sometimes research can be a competitive space, naturally, like it might need to be, but at the training level, we all need to be supporting each other to do really good work and to work together to learn. And that's the space that James Macaulay fosters, and I'm very lucky to be a part of it.
Thank you. Kerith, back to you. Obviously, you're, I think, lining up to submit your PhD in about another 6 months, 6-7 months, something like that. Where are you seeing, your future research career? Like, where to after PhD?
Yeah, that's a really, really good question, and is on my mind quite a lot lately. I think the value of doing a PhD means you're amassing a whole skill set outside of research and the particular field that you're working in as well. So I think I feel very privileged to be in the clinical space. Not many scientists and researchers get to work directly with clinicians, and I find that work very rewarding and very motivating. If I were to find a position, and it might be a position that I have to create for myself, that at the moment, being able to think in a diagnostic way that will actually help the families get an answer, but then also being able to use my research instincts and the flexibility that we have there to do some blue sky thinking around what is possible and how can we push these boundaries to learn more about the genome. I can really relate to Steve's analogy of the brain and astronomy. It's very similar for the genome. I think there are novel techniques that are still about to be discovered and brought into the clinic. And if I can be part of that process, bringing genomics, fully integrating genomics into Australian healthcare, then I'd be very, very happy to do that.
Am I right to think that, the two obvious pathways are kind of stay in academia or go into industry?
I think those are the main pathways. Obviously, academia is the one that we're baby academics. We're training to be full-fledged academics. But I truly believe that there are spaces in between. I think I have thrived finding those in-between spaces all my life. And so I'm hoping to be able to perhaps bring the best of both worlds together in a role that might be in industry, might be in academia.
I like that idea of creating the in-between spaces. Steve, what about you? I mean, you're past PhD, you're a fellow now. What's your ultimate ambition in terms of your research career?
Well, I've come from poverty, so my parents desperately wanted me to get a job that wasn't lifting bricks or something like that. And so at age 10, after watching some documentaries, I realized you could get paid to think. And so basically from that age, I wanted to become a professor. And ultimately, that's where I'd like to go. And it's as simple as that. I think I've been lucky that I sort of know the trajectory that I want to take. And, I'm on getting there. I've had my first few steps. Still need another 100,000 citations and a few more books. But that's my goal. I'd be a professor, and if there was something above that I could dedicate even more time to research, then I would.
Thank you. Hayley, I've got a few questions piling up on pain, so I might give you, I might combine a couple. So one question, is there any link in teenage anxiety, depression and chronic pain? And sort of linked to that, can chronic pain be a manifestation of bad things happening in a person's life? And if so, how do you address that?
Okay, so the first question, yes, there are links between depression and anxiety in youth and chronic pain. There's some evidence to suggest that chronic pain can lead to depression and anxiety, and then also in the other direction as well. So certainly as part of treatment, dealing with depression and anxiety in youth is an important aspect. One of the good things about the space I'm in about education is that it can help provide biological reasons for why it would be a good idea to engage in psychological services to try to improve pain and disability, depression and anxiety as a method of trying to improve your pain. And the second question, so what I would say to that is that what we understand about pain is that the brain will create pain if it assesses that there is a need to protect you. And one big part of the information that it will take in to do that will be what is happening in the body, the information that it's getting, but also it's going to take in any information it can. So if there are life events or stresses that could provide a reason to need to protect you, then that would theoretically give an argument for why pain could also be increased. So yes, yeah, a lot of therapies do look at trying to deal with anything within your life that might be driving up a pain response, driving up a need to protect, and that could be bad experiences from the past.
Thank you. Steve, I've got a couple of more technical questions here for you, again, which I might sort of combine. Question about whether neurostaining under MRI would help to gain more understanding of what's happening in cerebral small vessel disease.
Well, absolutely. And so the idea of staining for neurons within an MRI is only that's at the cutting edge right now. And so there's very few desperate research elements that are trying and merge those two together. And although not a very specific answer, it will, by its overlap, answer many, many questions in the research field and give insight into things we knew we could get insight into, but also insight into places we were unaware would get insight into. And so, although not a very specific answer to the second part of your question, it will reveal more and give us more information that we can use to develop an impact on anything, on understanding and treatment.
Thank you. And sort of slightly related question, for someone coming into the brain imaging world from a clinical background, would you recommend any particular resources for upskilling in neuroanatomy and brain function, particularly relevant for VBM and fMRI, which I'm not sure what they are, but you'll know.
Weirdly serendipitous that this has come up, because I'm just doing a paper right now with clinicians on not necessarily specifically upskilling them, but highlighting that a lot of clinicians aren't aware, not by any fault of their own, of the advancements in neuroanatomy that we have. We're actually still using some research from 50, 60 years ago in the clinical space for neuroimaging that, you know, labs like ours, my own and with George, have gone well beyond. And so we're highlighting that. And so I can tell you one resource straight away is Atlas of the Human Brain, 4th edition. That's going to really upskill you, because it's going to have not only the older, more classical Brodmann areas, which a lot of people are familiar with, but overlays them with a lot of the newer information we have, a lot of the newer functional information we have as well. And then there's a new functional atlas coming out probably in the next year or so that will merge those two together even more. There's a whole lot, actually. I'm getting lost in my head as it desperately wants to grab for more information, but Atlas of the Human Brain, Paxon is one of the authors, so is Mai. That's the best way to upskill. Where do you get
them, Steve? Where do you buy them?
Well, you can buy, they're published by Elsevier, but if you were to simply even just go to Google, because there's going to be a lot of distributors, and punch in Atlas of the Human Brain, Mai, Paxonos, you're going to bring up a whole bunch of distributors. I don't know how much it's going to cost, more than 100, less than 5. And it's the ability for it to overlay, superimpose the older archaic information with some of the newer stuff we have is the best way to make that translation.
Thank you. And Kerith, back to you for what will probably be our last question, I think. As a woman in science, right, and obviously I think you mentioned you've got 2 kids and so on. How do you find that journey? Is that particularly challenging? I know at NeuRA we do really well in terms of having a lot of women in science and fostering them in their careers, but is it just that much tougher? I think you're on mute.
That is a very tricky question. Thanks, Carole. Look, I think there's definitely a lot more awareness of the biases that are faced, not only by women, but also people of color in science. And I think there's so much conversation around that, that's really the first step. I think my experiences in particular have been generally fantastic, because I've had some great mentors and some great people teams that I've worked with throughout my career. Kids, it's always a juggle. There's no 2 ways about it. You have to be very firm with your boundaries, I think, which I've had to learn through this journey. My time with my kids is my time with my kids. And, when I'm at work, I have the privilege of them being at daycare so that I can concentrate on my work. I think there is something to be said, and I'm probably going to get told off when I go home about this, but there is something to be said about women, picking up sort of more of the emotional burden around the house. And we're just, naturally, I guess, poised to be the nurturers. And I think, you know, for example, today my son is sick, and I'm just, you know, really happy that my husband can stay home with him, and I you know, get to be here and discuss the science and discuss my journey. I think that's really important, coming back to that support system. But yeah, I think we're coming together as women in science, and we're definitely talking about this more, where I know that I follow a lot of you know, the global initiative program that's run by in London by our only woman Prime Minister, and she's doing some amazing work bringing out statistics to light about factors that, you know, affect women in science and women in leadership, and strategies that we can use to start to overcome those biases. So yeah, I feel much better equipped to be able to maybe confront those if I ever do now.
All right, thank you. Hayley, any quick last comment on that?
No, I think that was covered well. No, I think no, you've done a great job. It's, yeah, it can be a complex space to navigate. Yeah, I have had good support, and I've had really good female leads that I've been able to look to get some inspiration and some advice from, and that's been really helpful for me.
All right, thank you. Steve, any last comment? You can be honorary woman in this regard.
Well, I'm unfortunate. I haven't quite found my family yet. But I can point out that women, although there are minuscule differences between the male and female brain, we do know that females are better able to handle emotional strife within a familiar space. And I will stop myself there before I dare say anything else.
All right. Sorry for lobbing in that hot potato. But, all right. We're drawing near to the end of our time together. I should mention that we are going to be fundraising in this, our 30th anniversary year, for these all-important PhD top-ups. So please, if anybody's interested, do let me know. Also, join me in thanking Hayley, Steve, and Kerith for speaking candidly with us, and my thanks to our audience for your engagement and input. Please do look out for your follow-up email tomorrow, and I hope you'll join me again for the next NeuRAtalks. Bye-bye.